In our last video, we talked about chemical reactions needing energy added to their system in order for them to proceed, in order for them to actually happen. And the energy that is added to a chemical reaction so that it can occur is called activation energy. We also talked about how catalysts are substances that reduce the amount of activation energy a chemical reaction needs in order for it to occur. Enzymes, which is what we're talking about right now, enzymes are biological catalysts. They are proteins that catalyze each step of a metabolic pathway. So, metabolic pathways don't just happen because they require a lot of activation energy in order for them to occur. And enzymes come in and act as that catalyst and reduce that activation energy that needs to be added to the system. Enzymes will catalyze all sorts of biological reactions. All kinds of biological reactions are catalyzed by enzymes, and they're essential for life functions. They are, like I said, proteins. They lower the activation energy needed. So instead of needing this much activation energy, with an enzyme, I only need this much activation energy. And they bind to the molecules. They bind to what are called substrates, which are reactants, in such a way that bringing them together, that binding to those substrates, brings them together so that they can interact, or binding to the substrate allows it to break apart. So they are facilitating bringing molecules together or breaking a molecule apart. Like I said, substrates are the chemical reactants in an enzyme catalyzed reaction. Enzymes will physically bind to the substrate, so they will form typically covalent bonds to facilitate their reaction, to hold those substrates in place. And they do that at a location called the active site. It's just the location where the substrate attaches to the enzyme. Now, they are specifically shaped to the molecule that they are binding to. So enzymes are specifically shaped to their substrates. And it makes the enzyme reactions very specific. There's a lot of specificity in enzyme reactions. So typically one enzyme will only catalyze one reaction. All right. They are specifically matched, and each reaction has its own enzyme. So just to diagram it here, let's say we have an enzyme, and it kind of looks like this blue blob right here. And in that enzyme, you have an active site. So our blue blob is our enzyme. And that active site interacts with a substrate. Okay? And that substrate will fit into the active site. So if we draw our little blue blob again, it doesn't quite look the same. Ooh, that's horrible. Let's try again. We draw our little blue blob again. That's better. Our substrate will actually fit into that active site. And this forms what is called an enzyme substrate complex. And when that forms, when our enzyme binds to our substrate at the active site, that allows for the chemical reaction to occur. So essentially what we're doing here is we're decreasing the amount of energy because we're giving a place for that chemical reaction to occur, because we're giving a place for that substrate to come where it needs to be. All right? So the enzyme will actually contort or shape so that it interacts with the substrate better, so that the bonds can be broken or formed better. It provides an optimal environment for the for the reaction to occur. So if maybe the reaction needs to be slightly acidic or slightly basic, or maybe the active site needs to be polar in order to pull in a polar substrate, or nonpolar to pull in a nonpolar substrate. No matter what it does, whether it's bending or shaping or slightly acidic or slightly basic, they always return to the original state. So they'll pop out those products, and they're ready to go again. They're ready to take in a new substrate. So that is a characteristic of catalysts. Catalysts will speed up the chemical reaction, but they are not themselves changed. They don't change themselves. They only allow that reaction to occur faster. Now, we talked here about how they can contort or change shape so that they can hold that substrate in better. And that is called the induced fit theory, or the induced fit hypothesis. So we're talking about whether or not how, kind of like an enzyme hug. That is the most simple way of explaining this. So it is a dynamic interaction between the enzyme and the substrate. So the enzyme is physically changing to hold that substrate in place so that there is optimal bonding between them, so that they can ensure that that substrate will be able to go through the reaction it needs to go. It increases reaction capabilities, so it's going to be able to happen faster. So it's almost like when that enzyme gets that substrate, it is hugging it into place so that it can react. It is hugging those, maybe if we're talking about a reaction that is synthesizing something, it is hugging that A and that B into place so that A and B can come together and make that AB complex right here. And this is what it looks like. So in this case, we're showing where we're breaking apart a material, and you can see our active site right here, right? The substrate binds to the active site, and the active site, if you notice, it's a little bit more rounded when that substrate binds in, and it kind of extends into that substrate so that it can bind to the substrate better, and that allows for this facilitation of the reaction, and we end up with products. Now this can happen with synthesis products where we're building things instead of breaking things down as well. All right. Now, enzymes are going to continue to act as long as the local environment that they are within is friendly to them. This is kind of like this is the process of homeostasis. All right. As long as the temperature is just right, as long as the pH is just right, they're going to be able to react. Kind of a Goldilocks hypothesis here, where the porridge is just right. If the porridge is not just right, if the porridge is too hot or too cold, or maybe the porridge is too acidic or too basic, Goldilocks isn't eating the porridge. And it's kind of how proteins are going to act. Proteins are structural molecules. They have to have their shapes in order to function. If the environment in which the proteins are located in changes, it can cause the protein to lose its function because the protein can then lose its shape. All right? So it's called a process called denaturation. And it's when they lose their shape. And that means that the active sites can no longer bond to the substrate. So if it gets too hot, it's going to distort the shape of the active site so the substrate can't fit in there. Or if it's too acidic, it's going to distort the shape of the active site so that the substrate can't fit in. And if the substrate can't attach to the enzyme cannot work. Now, in most cases, denaturation can be reversed. All right, so if we return back to homeostatic ranges, if we get our temperature back to where it needs to be, or our pH back to where it needs to be, the protein will regain its shape and regain its function. However, it can be permanent. If the temperature goes too high, what it does is it changes the bonds between the polypeptide strand, and they can't go back to the way that they were before. Example of this would be an egg. If I throw an egg onto a frying pan and I heat that egg up to the point where the proteins in that egg denature, it changes the way the chemical bonds are between the polypeptide strand, and that egg is cooked. I can't uncook an egg. That egg is cooked for all time. All right, so it is irreversible. So some situations, a denatured protein cannot return back to its original shape. In most situations, they can return back to the original shape. When we talk about enzymes and enzyme activity, we have to talk about enzyme regulation. Living systems, cells, bodies, we have to be able to turn on enzyme activity. We have to be able to turn off enzyme activity. Sometimes it needs to be sped up. Sometimes it needs to be slowed down. So enzyme activity is regulated. And there's several different ways that enzymes are regulated. So the rate of the actual reaction is regulated. The amount of enzymes present to catalyze the chemical reaction affects the rate of the chemical reaction. So if there are a lot of enzymes present, it's going to be able to accomplish the job fast. If there's not very many enzymes present, it's going to take a lot longer to catalyze the chemical reaction. It's like when you're cleaning house. If you have a lot of housework to do and there's only one individual doing it, it's going to take a long time. But if 3 or 4 people lend a hand, pitch in, it doesn't take as much time to clean that house. So the amount of enzymes actually performing the reaction present changes the rate of the reaction. The amount of substrates for the reaction changes the rate of reaction. If there's not very many substrates to process in the chemical reaction, it's going to happen really fast. If there are a ton of substrates that need to be processed in the chemical reaction, it can take a lot longer. And the two of these are related. So if you have a ton of substrates, but not very many enzymes, you're going to have a slow reaction. If you have a ton of substrates with a lot of enzymes, you're going to have a faster reaction. The environmental controls, such as temperature, we talked about that when we talked about denaturation. Temperature and pH. If it gets too hot or gets too cold, the reaction is not going to proceed the way that it should. So temperature and environmental controls can affect how fast an enzyme can accomplish a chemical reaction. And then there's molecular control. Molecular control is when molecules are affecting the rate of enzyme function. We're going to talk about several different types of molecular control, but molecules affect the way the enzyme regulates. So let's first talk about competitive inhibition. Inhibition is slowing or stopping something. So if something is being inhibited, it's being slowed or it's being stopped. In this case, we're talking about slowing or stopping the enzyme reaction. Competitive inhibition is when a molecule that is similar to the substrate is competing for the active site. All right? So both the molecule, the substrate, and a competitive molecule are binding to that active site. If the substrate gets there first, the chemical reaction occurs. If the competitive inhibitor molecule gets there and binds to the active site, it keeps the substrate from being able to attach. And if the substrate can't attach, we can't have that reaction. It's very similar to my kids and our couch. All right? They both want to be able to lay down on that couch at the same, and it keeps the other one from being able to sit there. So if my daughter gets there first, she lays out, and my son can't sit there. But if my son gets there first, she lays down on the couch, and she can't sit there. So they're competing for that couch site, much like a substrate and a competitive inhibitor are both bonding to an active site. If the substrate gets there, our reaction goes. If our competitive inhibitor gets there, our reaction doesn't go. Another example is non-competitive inhibition. So this is when we're still slowing or stopping, but it's not through a competitive means. So a molecule will bind to the enzyme at a location other than the active site. So if we have our enzyme, right, and it's going to bind to a location other than the active site, and then it blocks the active site. So the substrate can't actually get in there. All right, it blocks the active site. It's let's say we're at a parking lot, and there's a truck there that has a bunch of really long boards sticking out of it, and they're not parked in the your parking spot, but the boards are blocking your parking spot, and you can't get in there to park. That's kind of how non-competitive inhibition works, where you have another molecule that blocks the active site. So it's not binding to the active site, it's just blocking it. Competitive inhibition, the other molecule binds to the active site. It competes. And non-competitive, we just block the active site. So this is a diagram that kind of explains that, where you have your enzyme right here, enzyme, you have your substrate, and then you have your active site. If everything is working great, that enzyme and the substrate will bind to each other, and we end up with our products, right? Everything's fine and dandy. Inhibition, you have an alternative molecule, all right? In competitive inhibition, that alternative molecule is going to block the active site for the actual substrate. So this little dude can't bind where he needs to be because this guy is in the way. So this is an example of competitive inhibition. Both of these molecules can fit in the active site, but the one that needs to fit in the active site, the substrate, can't get there because it's being blocked. That first molecule is bound there. All right. There's another type of enzyme regulation called allosteric regulation, and that is when a molecule binds to a location other than the active site, similar to non-competitive inhibition, but instead of blocking the active site, like we talked about with non-competitive inhibition right here, instead of blocking the active site, it actually changes the active site. So it's changing the active site, and it's going to change the active site in one of two ways. It can inhibit, where it changes the active site by this little block binding to our enzyme, changes the shape of the active site, and the substrate no longer fits in there. That's allosteric inhibitor. So the inhibitor binds to an allosteric site, a site other than the active site. It changes the shape of the active site, and the substrate can no longer fit. So we've stopped the activity of that enzyme. You can also have allosteric activators. And in an activator situation, the activator binds to the enzyme, and it changes the active site from a non-working active site to a working active site. So it turns the enzyme on so that that substrate can bind. And that's how it's going to whether turn the enzyme on or turn the enzyme off. Well, why would you want to turn an enzyme on or off? Let's say you have an enzyme that's producing a product and you have too much of the product. You only need a certain amount of product. You don't want to overload, or you don't want it to become toxic or something like that within the cell. So they turn the enzyme off to slow down that production, right? Or maybe there's a situation in the cell where they need a product, but they don't need it all the time. They only need it sometimes. So at the times at which the product is needed, the enzyme is turned on so that the product can be made. So allosteric regulation and competitive inhibition, non-competitive inhibition aren't the only type of enzyme regulation. There are also non-protein helper molecules that allow enzymes to function more efficiently. So they're not affecting the active site so much. They're not affecting whether or not the substrate can bind to the active site, but they're promoting optimal conformation and function. They're kind of like the Gatorade of the enzyme world, right? So supposedly Gatorade helps to promote optimal functioning because it's supposedly giving us all of these ions and things like that, salts and things like that to help us function, to help rehydrate us, help the athlete perform at optimal conditions, right? We also have molecules that help enzymes perform at optimal conditions. They are called coenzymes and cofactors. Coenzymes are organic molecules, which means they're made of hydrogen and carbon, and they will attach to the enzyme and help it function better. We get coenzymes from our dietary vitamins, so the food that we eat. We break that down, and those dietary vitamins are either going to be precursors to a coenzyme, so they're going to turn into a coenzyme, or they are themselves coenzymes. Cofactors are inorganic ions. So remember, an ion is something that has a positive or a negative charge, such as iron and magnesium, and those help our enzymes function better. So we talked about the ability of an enzyme to function correctly, the ability to inhibit or stop an enzyme. So let's talk about feedback mechanisms and metabolic pathways. So metabolic pathways, if you remember, is where we start with A and it takes several steps, several reactions for us to get to B. All right. At each of these reactions in a living metabolic pathway are going to be facilitated by enzymes. So that is a metabolic pathway. Okay, and if you remember back at the beginning of the first video where we looked at the metabolic pathway that allows for the breakdown of our macromolecules into ATP, it took several steps to get from our carbohydrate to an ATP, or to get from a lipid to an ATP. Now, sometimes we need to regulate that pathway. Sometimes we want pathways to be on, and sometimes we want pathways to be off. So feedback inhibition is a way that enzyme reaction rates can change depending on the amount of product availability. All right. So, for instance, we have this substrate right here, and it's going into this pathway, and we bind to enzyme A, and that gives us intermediate substrate A. Intermediate substrate A binds to enzyme 2, which gives us intermediate substrate B. Intermediate substrate B binds to enzyme 3, which gives us our product. We are here. Yay, we made it, right? That product will actually bind to enzyme 1. So this is enzyme 1, the same one right here. And it's going to change the shape of the active site, keeping that enzyme from producing more product. Now, if we need more product, it's going to leave, and the enzyme is going to be able to go down the pathway again. We're going to be able to move down our enzymatic pathway and create more product. So it helps to regulate cell processes. If we have a lot of a product, we can shut it down. If we need more product, we can turn it on. And it helps to regulate a lot of the things that are running our cells. All right, so we are done with our metabolism lectures. As always, if you have any questions, shoot me an email, send me a remind message, talk to me on Blackboard, but hopefully we have a better understanding of how enzymes function.